Here we have started with the first chapter of Standard 11, and that is cell biology. I think some time back, a picture of all these different types of cells was sent to you, and it was expected that you will identify these cells. Can you identify these cells? Some of you might have tried. Even now you can look at these, stop the video for a while, first identify the cells and then continue to observe the video, because this is a good pre-exercise. So once again I am suggesting, if you have not already sent the picture that was sent to you some time back, please stop the video right now and identify these pictures. Stop the video and identify these pictures and then continue. There will be a lot of fun. We must promote self-learning. Many of these cells are known to you, but it is quite possible that you are not remembering the names, and that happens. Let me tell you, there are so many different types of cells which make life, and what I have done is nothing. NCERT gives only 4 pictures of different types of cells. I am giving you only 21, and in nature there are thousands and thousands of different types of cells. What do you mean by types of cells? Types of cells will mean the different designs of the cells. Okay, assuming that you have identified them, try to identify. Okay, what is this? This appears to be a star-like cell. Well, the star-like cell is fibrocyte, and if you are not been able to identify it, it's quite okay, because you may not be knowing it is one of the types of connective tissue cells. Absolutely normal. If you don't know, it doesn't matter. But now you are trying to understand it now. Fibrocyte. Now number 2, this is a neutrophil. This is a white blood cell. It's a type of white blood cell, neutrophil, multilobe nucleus. Maybe that in your books, I think some of you may have had this kind of a diagram. White blood cells have a nucleus with different shapes. Number 3, nobody is going to make a mistake. You can call it as an amoeba cell or amoeba-like cells. It's not the irregular shape is possessed only by amoeba. There are many other organisms, many other cells which possess the irregular shape. Even this neutrophil or monocyte, all these white blood cells, although we are drawing them circular, basically while they are moving in the body, they are irregular, amoeba-like. Okay, so there are many amoeboid cells. Amoeboid means what? Amoeba-like cells, and typical amoeba, of course, is like that. Number 4, can you identify? Yeah, this is what is called as a phloem cell. And phloem cell, sieve tube. I will better write it as a sieve tube. Have you heard of sieve tube? Have you heard of phloem tissue? Phloem tissue has sieve tube. See, for example, this is a sieve tube. But there is one more cell which is accompanying, and that is called as companion cell. Companion cell. Companion cells and sieve tubes make the phloem tissue. They are tall, elongated cells, and the sieve tubes don't have a nucleus because there is cytoplasm which is moving through it, and food is being transported. And the companion cell is providing all the necessary help to the sieve tubes. Anyway, oh my God, this is something very different. You may or may not have identified it, doesn't matter. This is what is called as Phloem cell. Phloem cell? Yes, it's looking like a flame. Can you see? This is a bunch of cilia, and it flickers, and so it is being called as Phloem cell. This is an excretory structure in the platyhelminths. The tapeworm and the liver fluke. That's a platyhelminths group in which there are excretory cells which are called as phloem cells. The purpose of all these diagrams which are there is to make you understand that cells are of so many different types. Shapes also are so many different ones. Size and shapes, they differ a lot. Then come to 6th. This you may not have identified. This is very interesting. This is choanocyte. Wherever the word cyte is there, it means cell. Choanocyte. It has got a kind of a collar here. This is typically present in all these sponges. Do you know porifera sponges? Yeah, They have these cells, and there is a flagellum here, and with the help of flagella, the choanocytes make continuous generate water current, because you know the porphyrians are those who have water flowing through their bodies. Number 7, I'm sure you have identified it correctly. This is some algal filament, and so this is let us call it as Spirogyra or some similar cell, Spirogyra cell, which is Spirogyra has a linear arrangement of cells. It's a kind of a filament, and you have a chloroplast, even like chloroplast. So this is rectangular type of a cell, at least in the side view, and this is a spherical cell, you can say. Each cell has a different shape. This is a bird egg of a bird, or you can say it's an egg of a reptile, typical egg. Remember, egg is the largest cell. What is the size of the egg of a normal bird, the hen's egg that we have? Just count. You have eggs at home. Just find out the length. It may be hardly 3 to 4 centimeters, not more than that. Have a look here. This is the cell. This is one cell. Interesting. It is one cell. But understand, this is still not the biggest cell. The biggest cell, as most of you know, is that of ostrich egg, which is 15 centimeter, very long. Ostrich egg is 15 centimeter, and it weighs more than a kg. You can imagine, terrible. Ostrich egg and one cell. You don't need microscope to look at big cells like this. This is one cell. But then, of course, such cells are very rare. Egg cells are always large, and the egg cells of the reptiles and birds are very large. But egg cells of the mammals are, of course, small, because development takes place inside. We will now consider the 9th one. I'm sure you must have identified this as sperm cell. Very special case. It's a long cell. It has got a head, which has the nucleus, the chromosomes, the middle piece, and a long tail. How different this is from the egg. Egg is different, sperm is different. Even in birds, sperms are like this. Even in birds, the sperm will meet the egg and the zygote will be formed. Sperms of all animals are more or less similar. How different and how specialized is the sperm cell? Number 10, these are the meristematic cells, plant cells. Meristematic cells, they are isodiametric, they are cuboidal, and they are that way very tiny and capable of division, and they usually will occupy the tips of the stem, tips of the branches, tips of the roots. Meristematic cells. This is ciliated epithelium. Ciliated epithelium. Ciliated epithelium is very famous, and most of you know that there are cilia. This is an epithelial tissue. We had an introductory session on tissues 2 days back, animal tissues, and you know that epithelium has various types. There can be a squamous epithelium, there can be columnar, and there can be a ciliated epithelium. So cells are so different. They are tall and they have cilia. Coming to number 12. Well, this is actually a muscle cell, striated muscle cell, because it has got bands. So striated muscle cell. I hope close-up is being taken, and so you are able to observe. Striated muscle cell, long cylindrical. Striated muscle cells are long cylindrical, and if you compare 12 with 14, 14 is also a muscle cell, and this is smooth muscle cell, non-striated. Smooth muscle cell, that is non-striated. Striated, the skeletal muscles, and they are very long, and we are going to show you actually how to make a slide of the striated muscles when we are going to consider the muscular tissue. And smooth muscle cells are in the wall of the blood vessels, in the wall of the intestine, in the wall of the ureter. These smooth muscles are there. The shapes are so different. This is so different. This is cylindrical, and this is spindle shape. What do you mean by spindle? Spindle is something which is having broader in the center and tapering at the end. See, this is broader in the center and tapering at the end. 13 no one is going to miss, and that's very, very popular. Neuron. So, neuron, the nerve cell, probably one of the longest cell in our bodies is neuron. Neurons are quite long. Some of the neurons may be even few centimeter, and some of the very rare neurons are even 1 meter long. So this is long. Just like this sperm cell is long, the neuron is also long. So some cells are long. Which are the long cells? This one, this one, this one. So some cells are long, some cells are oval, some cells are spherical, some of them will be disc-like. 23. Red blood cell. It's a disc-like thing. Red blood cell. And so it's a, and just like a red blood cell is disc-like, there can be flat cells. 19. This is what is called as squamous epithelium. Squamous epithelium. Do not worry. If you don't understand everything, this is only beginning. Many of you may not be understanding. Let me tell you my story. When my father sent me to Pune in 1967, I was born and brought up at Jalgaon, and then I was sent to Pune for further education. And I was admitted to one of the great colleges then, and that was SP College Pune. First year I did not understand anything. Actually, I never wanted to go to science. But my father and my brother said, no, you have to do science those days. First year I understood no physics, no chemistry, no biology. But then I was alert. I said, if I have to take up this challenge, I must have patience. That's what I'm trying to tell you. Most of you are understanding what I'm trying to say, and some of you are not understanding. I am with them who are not understanding, because I remember my days of beginning of science. Science is always in the beginning likely to be difficult for some of us, and therefore have patience and see what happens. You have a great treasure of knowledge available through science. So, once again, we have 19 as squamous epithelium. This is squamous epithelium, this is ciliated epithelium. Although they are epithelial cells, these are flat cells, and the shapes are polygonal, and these are pillar-like cells with the cilia. Then I would suggest you to look at these cells, 16. I'm sure many of you are identifying these as the mesophyll cells. Have you heard of mesophyll? Mesophyll is a term. In your school books it is given that mesophyll is the green tissue of the leaves, and the cells are usually taller towards the upper epidermis. Maybe that some of you might have heard of palisade parenchyma. And don't worry, I'll simply say leaf parenchyma. So green parenchyma, I'll write right here, green parenchyma. What do you mean by parenchyma? Parenchyma is even 17. 17 is also parenchyma. This is non-green parenchyma. Where is parenchyma tissue present? Most of the leaf and most of the stem is made up of parenchyma. In the leaf, the cells are green parenchyma, and in the stem and the root are colorless parenchyma. So this is parenchyma. Parenchyma in the stem and the root are colorless. They have vacuoles. Even these have vacuoles, but here the vacuoles are quite prominent. These are quite large cells. Parenchymas are one of the largest cells of the plants. They may be 100 micron, 200 micron big, and I'm going to talk about the dimensions little later on. So this is parenchyma of the leaf, this is parenchyma of the stem. Parenchyma is one of the very common types of cells in plants. I would now select 15. I'm sure you have identified this. These are the kidney-shaped guard cells. You know stomach. Between the two guard cells, what is hidden is stoma. This is the the stoma is a minute opening on the leaf, and it is supported and regulated by what are called as guard cells, which are kidney shaped. The shapes, one purpose here is to know the variety and so also the shapes. So whereas this is flat like a tile, this is tall like a pillar, and this is now like a kidney shaped guard cells. Before I go ahead, I want you to identify, out of these total 22 numbers, I want you to identify something which is not a cell. I should have asked this earlier. Still a few things are left. I'll repeat my question. There are 22 simple structures which are shown here. Out of these 22 structures, please try to identify, out of the remaining which are left, which are not the cells. And this is something, this is not a cell, and this is also not a cell. Kaustubh sir, you are behind the camera. So can you tell us why the 20 and 21 are not cells? Then what are they?
Because they just have proteins and a nucleic acid.
So what are they then?
Viruses.
Oh, they are viruses, and viruses are not having any cells. Remember, so these two are not cells. So, Kaustubh sir, can you name this particular type of virus, a general thing?
Bacteriophage.
Bacteriophage. It's a bacterial virus. It is a virus that will be parasitic on bacteria. And this is an animal virus. HIV, for example, human immunodeficiency virus will look something like this. So 20 and 21 are living things. Viruses are living, no doubt, but they are not cells. So they are not cells. All the remaining are cells. This is paramecium 22. Paramecium everybody knows, and its shape is so different. It's like a slipper shaped. So paramecium. I think most of the cells have been covered. Very interesting. 24. I think because of the space limitation and my limited ability of drawing at 24, I could not draw in a better way. I'm going to show you a slide about it. Just remember that this is what is called as diatom. Diatoms are tremendous. Diatoms are impossible to believe. They have almost infinite different types of shapes and different types of architecture. Every cell is differently designed in diatoms. My God, I will be able to show you diatoms in one of the pictures little later today. So these are the different things that we have tried to take into account. These are all different types of cells, and cell biology is all about this. So look here. First lesson, cell biology, and you will have these two references in the NCERT book of the Standard 11, the chapter number 8, titled as The Unit of Life, and in the State Board book, chapter number 5, Cell Structure and Organization. The titles are different, but basically the material which is given in both the lessons is same. And let me tell you, I hope you have these books. If you don't have, please make sure that you have the NCERT Volume 1 and the State Board Standard 11. Because if you don't have books, there will be some problem. Of course, if you don't have them immediately, and if you are trying to get them, purchase them, if they are not available, okay, take some time, but then please make sure that you will have them. This is the lesson, cell biology, you can say cell science, and I have written it, this is for NEET. Why? Because we are all interested in NEET. NEET is one such ordinary examination. I will keep on saying it's not difficult at all. That's one thing. Biology scores will be very high. But learning biology is such a joyful thing. NEET is a very small purpose. Okay, learning biology for NEET? No. Learning biology for the big purpose, and NEET is a small purpose. You will automatically do it, but enjoy biology learning. So we will cover this in 8 lectures. Today's is the first. Every time the lesson will start with chapter outline. Please understand. This is the practice that we have been following all throughout these years for 11th and 12th. Every chapter begins with chapter outline. What is the advantage? The advantage that you come to know what you are going to learn. And while making this chapter outline, we have taken into account both your books. Both your books are having the same syllabus. Please understand, there is a lot of misunderstanding among students and parents that the syllabus is different. No, state board syllabus, all states of India have the syllabus exactly the same as the central board. Central board means NCERT books. So books are different, syllabus is same, and therefore be very comfortable. So whether you learn it from NCERT or state board book, you will get the same effect. In fact, those of you who have the books, you just go through the books. Go through this chapter 8 of the NCERT and go to this chapter 5 of the state board book. You will definitely understand that state board book is more richer, more better planned than the NCERT. So don't worry, learn the subject, whether it is NCERT or state board book, doesn't matter. You will definitely use both these books for all the chapters throughout the year. So actually, the chapter has some basic concepts which includes something like general aspects of the cells, common properties and common features of the cells, and these are all related to what is popularly known as cell theory. What is cell theory? Well, everything, all the life forms are made up of cells. I am going to talk of cell theory a little later on. So basic concepts which include general aspects of the cells, common properties or common features of the cells, and these are related to cell theory. So this is something we can talk about. And of course, you want to know about the prokaryotic cell. You know the bacteria, they are prokaryotic cells. Prokaryotic cells, most of you know, these are the cells without nucleus. You want to learn the ultrastructure. What do you mean by ultrastructure? Electron microscopic structure. So we are going to learn the prokaryotic cell, ultrastructure of the prokaryotic cell, and how the primitive prokaryotic cell then be compared with the eukaryotic cells. Because you know, eukaryotic cells are the cells of the plants, cells of the animals, cells of the fungi, and some of the protista, like the paramecium and euglena, and so on and so forth. You should know, you should remind yourself from your school books, prokaryotic cell and eukaryotic cells. Prokaryotic cells, primitive nucleus, actually no nucleus, and eukaryotic with a nucleus. Advanced cells are eukaryotic cells, and you will be learning the prokaryotic cell and then you will be comparing them with the eukaryotic cells. So this is going to be the first part. This is going to be the first part. We are going to learn this much today. Microscope is not given in the books properly, but it is very necessary to know the details of the microscopy, because without understanding the background science of the microscopy, how are you going to learn cells? Cells are those which are very minute. We have to find the reason why cells are so minute, and because they are minute, naturally you require microscopes. You know now the electron microscope. So we are going to learn a lot of electron microscopic structures. So you should know what is an electron microscope. But before you know the electron microscope, you should know what is a compound microscope and how does it work. And there are some other microscopes which are called as phase contrast microscope and this and that. So we are going to conduct a small session on microscopy. So these three together is part 1. So this chapter will be learned by us. You will be learning. We will also be learning. Teachers are always learners. We are learning, we are reading. So we will all learn together in 3 lectures. First lecture is this one, second is this one, and third is microscope. You can expect this. After that, the second part will start. The second part will be 5 lectures. Which are those 5 lectures? The entire second part is about eukaryotic cells. Because you know that most of the cells that we generally think of are the eukaryotic cells, and the eukaryotic cells are plant cells, animal cells, and the fungal cells. And you want to know the detailed parts, ultrastructure, electron microscopic structure of things like cell wall, plasma membrane, endoplasmic reticulum, ribosomes, Golgi apparatus, which is also called as Golgi complex or Golgi body, lysosomes, vacuoles, chloroplasts, mitochondria. Most interesting and important, nucleus with chromosomes, cytoskeleton, centrioles, cilia and flagella. Oh my god, so many parts of the cells. Again, I am trying to remind you of the levels of organization. Cells are made up of cell parts. All these things are better visible now because of electron microscope. Remember, nucleus can be seen without electron microscope. That is possible. Cell wall can be seen without electron microscope. But most of the vacuoles are of course very large, so they can be seen without electron microscope. But if electron microscope is available, then probably these things are seen better. As per your requirement of your learning, it is necessary that you know the electron microscopic structure. So it takes time because there is so much of research that has occurred, particularly 1950 onwards. In the last 70 years, so much of cell biology has been generated and some great books have been produced. This is one such book that we have, Essentials of Cell Biology. Remember, Essentials of Cell Biology is one such book we have, and many such books are there in the Learning Center library. We are proud and feeling happy to say that we have some good book collections, and although internet is being used, no doubt, I think there is no substitute for books. We have some other books, A Guide to Our Living Cell. A Guide to Our Living Cell. This book has been written by Christian de Douai, and he was the person who discovered lysosomes. Then there is much earlier edition of the Cell Biology by D. Robertis and others. It's a very good book of cell biology. And then we have another book about the cell biology. So we have many more books, but I'm not. They can be shown to you later on. Why books are important? Learning science, you have to have reference material. Are you going to refer all these books? No. Then why are we showing these things to you? To make you understand that as you go ahead in education, after 12, when you go in different branches of education, you will have to be independent. In the higher classes, in the medical college, in the engineering college, in the pharmacy college, nobody teaches. You, the students, learn on their own. And that's why they should be able to refer books. So they should know that there are books. And books are very important for us. And then you can become independent. So there are lots of good books, and we try to refer them before we prepare for the lecture. So all these are ultrastructures, and they will take 5 lectures. So all these will be divided into all these cell parts will be discussed in 5. You can understand that for on a given day at the most 2 or sometimes 3 parts will be explained. So there are so many parts. So 5 plus 3 will make 8. So we are now at the initial consideration of this chapter. This very popular term like cell theory. What do you mean by cell theory? What is cell theory? Oh my god, it's a very simple thing. Cell theory is what? All life forms are made up of cells. So you may say, oh, if that is so simple, then why to talk so much about cell theory? There is a reason why we must talk about cell theory, simply because it has not been suddenly proved that all life is made up of cells. There is a long history of research. Lots of original discoveries have been done by these people. These are stalwart people in the field, and they have contributed in the last almost 300 years. They have contributed their researches to make a firm statement that everything is made up of cells. It was not that easy. Today, everybody knows. Even a first or second standard student also knows what are cells. But then it took 300 years to finally establish that everything is made up of cells, all life is made up of cells. So these are the basic discoveries. And because of their discoveries now, certain modern concepts have been developed. The modern concept is what? Cell is a metabolic unit of life. Cell is a genetic unit of life. Cell has totipotency. And to all these things, there are some exceptions. I mean to say that the cell theory is something which is a background for the study of the cells. Yeah, cell theory is that way historical. But history of science is very important. When you learn atom, you always learn the history of atomic research. You know this? You know this very well. So the different people in the last 150 years started suggesting more and more views about the atoms, and now you know what atoms are today. Just like in physics, you learn the history of atomic research. In a similar way, in biology, you learn the history of the research about cells. So cell theory is something which is ordinary. Yeah, I understand. It means how it is established that cells are the units of life. Cells are units of life. It's very clearly mentioned here. All these terms are already mentioned in the soul, in the notes. Cell is a metabolic unit of life. Cell is a genetic unit of life. Cell is having totipotency, and so on. If you go through the board books and the NCERT, there are some additional points like living things are made up of cells or cell products. Sometimes it is mentioned that living things are made up of cells or cell products. Why it is being mentioned cell products? Because think of the bone. Bone has cells, no doubt, but the cells of the bones are kept away from each other because it's a connective tissue, and between the cells there are layers of matrix. Have you heard of this word matrix? Matrix is which is non-living substance which is produced by cells. If suppose the bone was made up only of cells, then it would be very soft because cells are soft, but bones are hard. So what is the hardness? What is the cause of the hardness of the bone? The cause of the hardness of the bone is the salts, the calcium and phosphorus salts, magnesium salts, and salts make the bones hard, and all these salts are secreted by cells. That is the reason why in your books you get statements like this, that living things are made up of cells and cell products. There is one more statement that is made, and actually it is suggested by this great scientist Rudolf Virchow, about all these scientists I am going to talk. Omnicella acella. Omnicella acella. You will get this statement in both the books. What is the meaning of these things? The statement, which is a Latinized wording, says that cells are formed from pre-existing cells. That means cells divide. But it was not clearly known till 1855. Understand, 1855, almost 170 years over. It was not possible to know that cells divide. So somebody wanted to clearly point out by experiments and observations that cells divide. And the statement comes up, omnicella acella. What does it mean? The cells come from pre-existing cells. What does it mean? That the cells divide. Then there is a statement like cells are functional units. What do you mean by functional unit? I am living. I am living because if most of my cells are living. I am saying most of my cells because some of my cells are dead. All the topmost cells of my skin are dead. Some of the red blood cells in my body are also dead. It's okay. But majority, 99% of my cells are living. So every living organism is living because the cells are living. You understand what do you mean by unit? What is the unit of measurement of a scale? Centimeter. The regular scale that you are using, what is unit? Centimeter, or at the most you can say millimeter. So just like centimeters and millimeters are units of measuring length, in a similar way, what is the measure for life? Measure for life is cells. What do you mean by that? It means that if life exists, it will be measured. And what is the measure? Cells. It means if cells are existing, life is existing. That is the meaning. It's not about the actual dimension. It is about it's a proof, if cells are observed, then it is life. The cell may be one, then it will be unicellular. Cells may be many, in that it is called as multicellular. And from that point of view, it is said that is a functional unit. Every cell independently works because it has got a nucleus and it has got so many cell parts. And the cell is almost always self-sufficient. It has got its own existence, and that is how it is a functional unit. There are some more things. The cells have unit membrane. What is the concept of unit membrane? We are going to learn all this in details in the lesson. You have heard of the cell membrane, whether it is bacterial cell, that is prokaryotic, or whether it is a eukaryotic cell, that of animals or plants. Please understand, membranes are very important. If you think of the membrane of the mitochondrion, membrane of the Golgi complex, membrane of the chloroplast, membrane of the nucleus, all these membranes have similar structure. I think in your books you had electron microscopic structure of plasma membrane or cell membrane. You may have understood it, or you may not have understood that time. But one thing, whatever is the ultra microscopic structure of a plasma membrane is the same which is the ultra microscopic structure of the chloroplast membrane, which is the same as is the ultra microscopic structure of the mitochondrial membrane. Ultimately cell is a complex design of membranes. What is a membrane? Membranes are sheets, and the sheets have, I think I'll just remind you that thing, these membranes or sheets have two major chemical components. Do you remember which are those? Lipids and proteins. Have you heard of these things? Have you read of these things? Do you remember? If not, don't worry. From my side, I will say you don't worry. You always be with us. Focus on the lectures. Repeatedly listen to them and see what happens within 1 or 2 months. You will be very comfortable in biology. So, what is the unit membrane concept? That there is a membrane which is surrounding the cell, and the same membrane, same design is available all inside. All the endoplasmic reticulum membrane, all the membranes of the Golgi complex and nucleus and mitochondria and chloroplast, absolutely same design. That's how cells have unit membrane. What does it mean? That all membranes outside and inside of the cells are same. That is the concept of unit membrane. Metabolic processes occur. I'm going to go into those details later on. Metabolic processes. Metabolism is chemical reactions. And every cell, whether it is a bacterial cell, or whether it is a neuron, or whether it is a cell of a leaf, or the cell of my eye, or cell of a muscle, lot of chemical reactions are going on. Hundreds and thousands of chemical reactions are going on. And that's how metabolic process occur, and a cell is that which has metabolic process. You don't get metabolic process outside. Non-living world does not have metabolic process. Non-living world doesn't have cells. Non-living world doesn't have membranes. Non-living world doesn't have chemical reactions, metabolic process. And about genetic control and transcription and translation, I'm going to talk. I mean to say, when now coming to writing answers, because question will be there, write down common properties of cells, because we have decided that we will be taking into account common properties or common features of the cells, and they are related to cell theory. When we are talking about these things, are we talking of cell theory? Yes, all this is cell theory only. This is about the research. This is about the general conclusions. Because of the studies of these people, now we can say that these are the characteristics of cells, properties of the cells, and the properties of the cells are a product of research, and that research is cell theory research. Cell is a metabolic unit of life. Listen carefully. I will now add a few words here. Metabolism. What do you mean by metabolism? Metabolism are chemical reactions in a cell. A given cell appears very simple. I can simply draw the outline of a cell and show a nucleus. But when you think of this cell, you be very minute. You become a very small particle and enter in a cell. And you will find that the cell is very busy. Hundreds and thousands of chemical reactions are going on. Probably you know that there are what are called as enzymes. Do you know what are enzymes? Enzymes are catalysts. Have you heard about catalyst? You know them. Catalyst is something which speeds up the reaction. All metabolism in the cell is possible due to enzymes. Every different chemical reaction will require a separate enzyme. If there are 1000 chemical reactions taking place inside, you can expect there are 1000 different types of enzymes. My God, so cell may be very minute, but it is excessively busy. It has got thousands of chemical reactions taking place, and those are very fast. You know, when chemical reactions occur outside in the test tube or beaker in the laboratories, they are usually slow. But when the chemical reactions are taking place in the cells, they are extremely fast. A particular chemical reaction which is occurring to the order of 10 raised to 1 outside, here it will be 10 raised to 7, 10 raised to 10. Terrible, very fast reactions. Please understand, if these reactions in the cells were slow, life wouldn't be existing. No cells would be existing, I won't be existing, you won't be existing. Why the cells are able to survive? Simply because thousands of chemical reactions are occurring very, very fast, and now those chemical reactions are of two types. Some of them are anabolic. What do you mean by anabolic? Constructive. Synthesis. New materials, new molecules are being produced. Small molecules come together to form large molecules. I can give you one example. Amino acids, have you heard of them? Amino acids are tiny, small molecules. Amino acids are small molecules, and they get joined together, and that forms a protein. I think this much you know. This is an amino acid, one amino acid, one amino acid, many amino acids, very tiny molecules, amino acids, and they get joined together, and that structure which is formed is called as a protein. This is anabolism. Similarly, glucose molecules are small. All glucose molecules get joined together, and a chain of glucose molecule is produced. And what is that chain of glucose molecules called as? The chain of glucose molecules will be called as cellulose. The chain of glucose molecule can also be formed called as glycogen. The chain of glucose molecules can also be called as starch. So the glucose molecule can get joined to each other in different ways, different fashions, and accordingly it will be either cellulose or starch or glycogen. So this is anabolism which is going on. But then there is one more thing, catabolism. Catabolism is what? Reverse of anabolism. Anabolism is synthesis, joining together. Catabolism is breakdown. Suppose this is a protein molecule which is produced. It is produced by joining of amino acids. Now you will break, these bonds get broken, and each amino acid will get separated. If each amino acid gets separated from a protein chain, what we will call this as? Catabolism. So, anabolism and catabolism continuously taking place very fast in a cell, and that is a concept of the cell as a metabolic unit of life. My God, if metabolism stops, no life possible. We are going to learn a lot. There is a special branch of biology that deals with metabolism, and you should have heard of that, and this is biochemistry. So biochemistry is a branch of biology which deals with metabolism, and you will have some biochemistry lessons to be learned in 11th and 12th. So you are going to learn metabolism. I'll just give you two examples. Photosynthesis in plants is biochemistry, and it is anabolism because glucose molecules are being produced. How glucose molecules are produced? They are being produced from carbohydrates. Glucose molecules produced from CO2 and water. CO2 and water are simple molecules. They come together to form a glucose molecule. And those glucose molecules get joined to each other to make starch molecule. That is photosynthesis. So there is anabolism. Think of the respiration. Most of you know respiration is catabolism. Why? Because the glucose molecule is broken. Glucose molecule is broken into carbon dioxide and water. That is respiration. It means that you are going to have a lesson on photosynthesis, you are going to have a lesson on respiration and some other aspects of cell life, and that is going to be biochemistry. Cell is a genetic unit of life. What do you mean by genetic unit of life? Because it is being mentioned here also, genetic control. You know this very well now because cell has a nucleus, and you know the nucleus has chromosomes, and the chromosomes are made up of, I'll repeat, cell has a nucleus, almost all cells have nucleus. Nucleus has chromosomes, and what are chromosomes made up of? DNA, the great molecule, being called as a master molecule of life. So the DNA has genes. I think most of you know this, that in the nucleus there is DNA, chromosomes and DNA is a long molecule, and the parts of the DNA are called as genes, and those genes control the working. If suppose there are 1,000 reactions taking place, there are 1,000 enzymes. If the 1,000 reactions are taking place, there are 1,000 enzymes, and each enzyme is controlled. Each enzyme is being produced by a separate gene. There will be 1,000 enzymes for 1,000 reactions, and to produce those 1,000 enzymes, there will be 1,000 genes. I can write it like this. 1,000 genes, 1,000 genes will produce 1,000 different types of enzymes, and 1,000 different types of enzymes will regulate 1,000 different types of reactions. And these reactions which are taking place will make the cell alive. So the life of a cell will depend on the thousands of reactions, and those thousands of reactions will be controlled by thousands of enzymes, and thousands of enzymes will be controlled by thousands of genes, and thousands of genes are nothing but pieces of DNA. Pieces means the segments of DNA. Wonderful. And you know this very well, the genes which are part of the DNA and chromosomes, that is the genetic unit. So every cell without fail is a genetic unit of life. Then some of you may be saying, no, red blood cells are without nucleus, so what about that? Yeah, I can provide you that explanation also. Red blood cells, when that is being designed in the bone marrow, in the beginning it has nucleus. So whatever enzymes are required and whatever reactions are to be carried out, right in the beginning, when the nucleus is still present, all those products are ready. And later on the nucleus is removed. Once the nucleus goes out, usually there is no genetic control left. That doesn't matter. But then such cells like red blood cells have a short life and their purpose is different. So those are only small exceptions. But otherwise, whether it is a paramecium or whether it is an amoeba, which is a eukaryotic unicellular organism, or whether it is a neuron of my body, or whether it is a liver cell, or whether it is the cell of my stomach, all organs, or whether it is a cell of a leaf or a root or a stem, all cells have nucleus, and nucleus has DNA, and parts of the DNA are called as genes, and genes are mostly regulating production of enzymes, and these enzymes regulate reaction. It's very important. This is what is genetic control. So every cell is a genetic unit of life. So metabolic unit and genetic unit, they get related. Are you understanding how the metabolic unit becomes related to genetic unit? There's a special science of this kind of working, and I'll call this as gene science. I can call it as gene science. Biotechnology I can say, but I will try to avoid that word because biotechnology is application of this knowledge. You can simply say DNA science. I can say DNA science. That's quite okay. So DNA science is a big branch of biology which has come up now, and you will have 2 lessons about the DNA science and to understand that it is a genetic unit of life. However, many of you must be expecting that when we are talking of cell as a genetic unit of life, why shouldn't we use the term genetics? If cell as a metabolic unit of life is related to biochemistry, then you may expect cell as a genetic unit of life should be related to genetics. You can, but genetics, typically of the Mendelian type genetics, the classical type of genetics, is probably not related to directly to the cells, because Mendel worked on pea plants. And many, or when we are talking of human genetics, we are generally talking of inheritance of characters. It is true that ultimately explanation has to be found at the level of cells, explanation has to be found at the level of nucleus, explanation has to be found at the level of chromosome. That is true. So it becomes then possible to talk of the ultimately of the DNA. So when we talk of the cell as a genetic unit of life, we talk of the nucleus, and when we talk of the nucleus, we talk of the DNA. At the most, you can say modern genetics. So you may think that cell as a genetic unit of life will be studied under modern genetics, just like the metabolism is studied under biochemistry. Cell has totipotency, a very interesting term, and I'm sure you are going to use this term many times during your 2-year biology course. Totipotent. What do you mean by totipotency? The totipotency means the ability of the cell to give rise to the entire organism. Yourself, myself, we started our development from a single cell. And now, the entire organism has been produced. 9 months, we are in our mother's uterus, and the single cell, which is of course zygote, gives rise to the baby, and this single cell has all the necessary record of making the human body. A zygote of human will form human, a zygote of fish will form a fish, a zygote of elephant will form an elephant, a zygote of a mango plant will form a mango plant. It means that the zygote which is formed as a product of fertilization, sperm cell and egg cell, the genes of the sperm cell and the genes of the egg cell come together to make the zygote. Everybody knows that. Those genes have the ability to form the entire organism. This property of the cell is called as totipotency. Is the zygote only cell to have totipotency? Is zygote the only cell to have totipotency? No. Actually, there can be some other cells which can be totipotent. Have you heard of what are called as stem cells? Have you heard of stem cells? I think most of you have heard of stem cells. And stem cells are those cells which remain in the original simple embryonic form. I've used the word embryonic. In every organ of my body, in the heart, in the liver, in the brain, in the lungs, in the intestine, there are some cells which are in the original form. Why? Because these cells can be converted into the regular cells. A great example is that of the bone marrow. Bone marrow is a very active tissue, because in the soft part of the bone, the spongy part of the bone marrow is there, red bone marrow, and there are a lot of stem cells, and from those stem cells all the different types of blood cells are being produced. So stem cells are undifferentiated. What do you mean by undifferentiated? Undifferentiated means they remain simple. Why they remain simple? So that they can form all the different cell types. So this is a totipotent cell. Of course, the totipotency of zygote is more original, more stronger. So zygote has the capacity to form the entire organism. But a stem cell in the bone marrow will not form entire organism. Usually not, cannot. Technology may make it possible later on. A stem cell in the bone marrow can form different blood cells and so on. So that is totipotency, which has got different levels. Plants have more totipotency than animals. Can you tell me which are the totipotent cells of the plants? Do you remember the plant tissues? Can you name a particular plant tissue because of which the plants can continue to grow throughout life? Yes, some of you have remembered meristematic tissue. At the tips of the roots, tips of the shoot, there is meristematic tissue, and because of that, new cells are continuously being formed. So meristematic tissues of the plants are a very good example of totipotency. So totipotency, what is totipotency? It's the ability of the cells to get converted into the entire organism. That's the broadest sense. But even if you expect that the given cell will be converted into some other tissue types, even that is called as totipotency. There are different levels of totipotency. I think we are not going to talk about it now. We will take into account the different aspects of the totipotency, pluripotency, multipotency. I think this is not the time to talk about all that. But yes, cells do have totipotency. People now in the field of cell biology, and the scientists are trying to make this possible, that any cell of our body, not just zygote, any cell of our body, suppose you take a liver cell, suppose you take a kidney cell, suppose you take a muscle cell, from that entire human body should be produced. Oh my God, you don't expect a liver cell will give rise to an entire human body. They say that whatever is the genetic information, in the nucleus is enough, that should give rise to the entire body. Because the zygote has a nucleus, and whatever the genes in the zygote, the same genes are there in the liver cell. Suppose this is a liver cell, and in that case it should be possible to get an entire organism. Well, that may not be immediately possible, but they are thinking of this, and some of those trials are being taken place. So there is a lot of curiosity about all such things. So that is the issue of the totipotency. So although the cells have characteristics like there is a typical organization, eukaryotic and prokaryotic type, there is a membrane which have got constant structure, inside membranes, outside membranes, then the metabolism and genetic control we have talked about. About transcription and translation, I will be talking about little later on. But even these words have come up in your school books up to 8, 9, 10. And I am going to hold separately explanation of these two terms, transcription and translation. These are the things because of which the genes are able to produce enzymes. When, suppose there are 1,000 genes which are inside the nucleus, and they give rise to 1,000 enzymes. All these enzymes are proteins. Are you aware? Have you heard that all enzymes are proteins? If not, now listen to this. All enzymes, almost all enzymes are proteins. And this particular process of the genes producing the enzymes, the DNA, genes means DNA, controlling the protein, DNA controlling the protein, and this particular process is divisible into two clear-cut segments, transcription and translation. We will be talking about this. So transcription and translation is a part of the genetic control, and genetic control is this, genes controlling the enzymes. So all these things are common to most of the cells. There are some exceptions to cell theory. Viruses are not cells. Interesting. Viruses are not cells. They don't have cell parts, nothing, and they are very, very minute. But then they are definitely considered living because they are able to reproduce. Prokaryotic cells. Why prokaryotic cells sometimes will be considered as exception to cell theory? Bacterial cells. They don't have nucleus. Generally, we are trying to say that cells are those which have nucleus. Cells are always with nucleus. That is usually the statement of the cell theory. But then there are some cells without nucleus, like the bacterial cells. So that can also be considered as an exception. There are some tubular cells of algae and fungi. There are some fungi which have you heard the word hypha, a tubular structure. Or some of the algae, primitive algae, they are like a tube and they don't have nucleus. Or sometimes they have many nuclei. Either they have no nuclei or they may have many nuclei. In that case, that is also an exception. Usually, original cell theory tries to say that one cell, one nucleus. But suppose there are many nuclei in a tube, no nucleus or many nuclei in an algal tube or a fungal tube, then it is something exception to cell theory. So I don't think you have to be very serious about exceptions. Exceptions are there always, and exceptions. In fact, whenever the exceptions exist, that means the original theory becomes strong. There is always a statement which is made like this: every exception is good for the original theory. It means if this is absent here and it's an exception, that means all the remaining cells must have all these things present automatically. So don't worry about these exceptions. So when you are writing about cell theory, the questions will be like this. The questions will be loaded after this lecture is over. You will be asked the questions, write down the properties or characteristics of the cells. Just for 1 or 2 marks, explain cell as a metabolic unit of life, explain cell as a genetic unit of life, write a note on totipotency. So such questions will be there. So what we have learned just now is a part of the cell theory. Why I am saying part? Because this thing I have not explained this. Because this is something which is the original research about which there is some serious comment I have to make. But we have generalized the modern concepts. What do you mean by modern concepts of cell theory? Whatever is known to you. When we are talking of the DNA, we are talking of the modern times. So this and this is same. This and this is same, but many times in the books they are making a list of this kind. In the state board book you will get a slightly different list. I am just trying to make this more broader. So all these concepts which are here are being made broader. I think what you need nowadays is not to mug up the answers. Up to school, probably, maybe that your teachers used to tell you that learn this much. Learn manje path kara. A very wrong meaning of learning. Learn karunya, manje path karunya. Horrible. Learning is not mugging up. What do you mean by learning? Learning is thinking. सगळे अर्थ बदलून घ्या. Learning is thinking. लर्न करूया म्हणजे विचार करून या असं त्या टीचरला सांगायचं असतं आणि तुम्ही पाठ करून येत. नो क्रॅमिंग, नो मगिंग अप. इट्स ऑफ नो यूज नाव. यू हॅव टू अंडरस्टँड द सब्जेक्ट्स. You have to understand the minute details of biology, physics, chemistry, maths. Then only you will survive. This age of competition, this age of science where there are selections possible, you need to understand the subject. So please make sure that you are understanding. Don't mug up. काही जण फक्त पाठ करतात त्याचा काय उपयोग नाही. But you should write. Biology will have to be learned by listening carefully, observing nature around, and writing. We are going to suggest you 3 notebook system. But now presently, because the first lesson has started, make sure that you are writing. What to write? You can make your own notes. तुम्हाला माहिती का नोट्स कशा काढायच्या? नोट्स काढायच्या काय? तुम्हाला जे समजतंय ते लिहून काढायचं. One thing, board writing as it is you can copy. That is one step. Board writing as it is you can copy. That also will provide you a good practice of writing. Then additional points you can add to that original thing. Suppose additional points, I said something about metabolic unit of life, I said something about genetic unit of life. Those things you can add. You can write, you can think, you can make your own statements. So please try to make notes. See, 11th, 12th is a very, very important stage of learning because after 12th, I'll repeat. Magashi मी तुम्हाला सांगितलं. After 12th, nobody teaches. Nobody will teach you in medical college and engineering college. ते भरभर सांगून मोकळे होतात. You must prepare yourself for self-learning. And that means you must understand things. You should be able to write paragraphs. You should be able to put proper comments at proper places. तशा तर नोट्स तयार करा. म्हणजे बोर्ड रायटिंग करून घेतलं असेल, इव्हन तुमचं चॅप्टर वाईज जे आउटलाईन आहे ना ते पण तुम्ही लिहिलंच पाहिजे. इनफॅक्ट ऑल द स्टुडंट्स ऑलवेज स्टार्ट विथ चॅप्टर लाईन आउटलाईन. आम्ही वर्गात तर त्यावेळेला रेग्युलर वर्ग होतो त्याला वेळ द्यायचो की चला हे लिहून घ्या आता, मग आम्ही 10 मिनिटं थांबायचो. Then they used to write the chapter outline. Now we don't need to wait because these things are with you. So you will all start, always write everything that is written here, should write. Then go for the cell theory. And then certain additional points I have told you. So what is to be done? Copy down everything. and then add additional comments. Lot of work. We are going to tell you that you must study biology at least for 2 to 3 hours a day. Physics, chemistry also will require a few hours, including the online learning timing. It means you have to be busy 8 to 10 hours. Are you going to be busy? Now most of you have physics and chemistry also bigger, and probably maths also. You will have 8 to 10 hours of work. When the regular classroom teaching is there, even that time students are busy like this. They have classes for 1 hour, 2 hours, and also some homework. It means you have to be busy. आता काही सुट्टी वगैरे चालू नाही आहे तुमची. Please understand, you have to be serious. Whether it is offline, that is ground level learning, or whether it is online, every time it all depends on the sincerity of the student. So please make a good effort. I'll come to certain other additional points, particularly about the original discoveries, after a pause. We have now tried to understand some basic concepts about cells. The cells may be of different shapes and different sizes. Dimensions I have not yet talked about. I will be talking about the dimensions little later on, but we have tried to take into account what are the common properties of the cells and in what way those common properties can be discussed. But what is now left is this much part. So I am talking now about the original discoveries. Great people like Robert Hooke, Leeuwenhoek, Robert Brown, Schleiden and Schwann, Ralph Virchow, Valdier, Huxley, and many more. Robert Hooke, 1665. Imagine the time, 350 years back. My God, that was quite old time. I mean, the world was very different those days. Think of the 1665 situation in India and in Maharashtra. Aurangzeb was in full swing. Mughal Sardar, Mughal Emperor, and Chhatrapati Shivaji was at the best of his helms for fighting out the Mughals, 1665. That is Shivaji Maharaj time I am talking about. And that time, Robert Hooke, for the first time, tried to tell the world that he took a section of the cork, the plant, bark, and then not bark, the cork, the outer skin, and then this showed that it is made up of compartments, which he called as cells. A little later on, Leeuwenhoek, we are going to see the pictures. He was again a person who could show that there are cells like sperms, red blood cells, bacteria. He was able to show them with a simple microscope. Robert Hooke was able to make a microscope, and Leeuwenhoek also made a very simple microscope. Robert Brown, little later on, much later on, 150 years later on. Robert Brown, Schleiden and Schwann, and Ralph Virchow, all these were in the time of Darwin, because most of you know more Darwin. So when Darwin was busy with the research on evolution, Robert Brown was busy with understanding cells. Schleiden and Schwann, these are star marked because the cell theory mostly is credited to these two people, botanist Schleiden and zoologist Schwann. I'll talk about this little later on. Rudolf Virchow, he was the one to say that new cells are formed from pre-existing cells. And Valdier, he was the one to say that chromosomes exist, and he also was the one to say that mitosis, the cell division begins. I mean, the basic research of this cell division was at least initiated by Valdier, because he was the one to see for the first time under a microscope chromosomes. Please understand, this was much older time. Robert Hooke and Leeuwenhoek, 17th century. Robert Brown, Schleiden Schwann, Rudolf Virchow, and Valdier, 19th century. This is 19th century. 1888, 1855, 1839 is 19th century. 1665, 1683, this is all 17th century. Huxley is in the 20th century, and he worked right up to, I think, 1974. And so that was 20th century, so no year is being written here. How much it is important to remember the years? To be very frank, not necessary. then don't ask me the question, NEET मध्ये आलं तर काय करायचं? This is the weakness of mind. If everything you want to relate to NEET, your mind is weak. And with a weak mind, you can't clear NEET. NEET साठी ना तुम्ही NEETची भीती काढून टाका. हे येईल का, मग कसं होईल, मला हे लक्षात राहिलं नाही तर कसं होईल, असं सगळं सोडून द्या. Learn science. So if you remember the years, it is good. If you don't remember, it is very good. मी काय सांगतो लक्षात घ्या. If you remember, good. If you don't remember, very good. Relax राहा. Most important thing to be successful in education and life is remain relaxed. You will remember it. It's okay. फार असं खूप प्रयत्न करून लक्षात ठेवू नका. आपोआप लक्षात राहील. But these are great people, remember. And wonderful thing, Robert Hooke and Leon Hooke, both of them were in a different field. Robert Hooke was a great physics person. You know, Robert Hooke and Newton were contemporaries. That means their times were same, and actually they interacted with each other. Their research was shared. And Robert Hooke was not a biologist, he was a physics person. There are certain special elasticity rules which he framed. Elasticity is a part of the physics. But then he also was a microbiologist, not a microbiologist, he was a cell biologist in the sense he was trying to use the lenses. He constructed a microscope, and in the PowerPoint presentation you will see his microscope. Leon Hooke was a merchant. He had some business at home, but as a hobby he did all this. Robert Hooke had some formal education at least. Leon Hooke had no formal education. So he did not go to any particular school, college, but then he said that he had a lot of interest in, craving for science. He wanted to know. So wanting to know is curiosity, and that curiosity should take you to science, not because of NEET and JEE. NEET आणि JEE या सारख्या अंमलाखाली तुम्ही राहू नका. शिकण्याची ओढ आणि आवड ज्याला असेल तो कुठेही NEET आणि JEE काय सगळीकडे जगात सिलेक्ट होईल. ते फार महत्त्वाचं आहे. I will repeatedly say this. Don't study for exams. Study because you are interested. And exams will be automatically done by you and you will get selected. So, understand, Leon Hooke's name has to be every time mentioned with cell biology because he did it a pioneering work. He was the first to observe bacteria. He was the first to observe the red cells, minute. He was first to observe the minute other organisms in dirty water. Robert Brown was a scientist in the sense he was of course busy with many other branches of science. He was also a guide to Charles Darwin, and Robert Brown is also known for Brownian Movement. Have you heard of Brownian Movement? You know, particles, they keep on moving in the solution. Particles in a solution keep on moving because of their free energies, and that is Brownian movement, and it can be observed. If pollen grains are put in water and a drop of water with the pollen grain is observed under a microscope, pollen grains will show movement. Brownian movement. Schleider was a botanist and Schwann was a zoologist. Robert Hooke, British person. Leon Hooke, Dutch person. Robert Brown, British. Schleider and Schwann, German. Rudolf Virchow, German. Rudolf Virchow, actually Schwann was a zoologist and a physician also. And Rudolf Virchow was again in the field of biology in addition to other fields, and he was the one to use microscopes to observe that cell division can occur. And then Wilder was an anatomist, and he was the one to say that the chromosomes exist and so on and so forth. So actually these are all very high grade figures. When we are mentioning them under this lesson, we are trying to make the image small. Please understand, it is only a side reference. Their credit to cell biology is a side reference in their own fields, again different fields. Many of them were paleontologists, many of them, most of them of course wrote books, many of them were also busy in politics those days, and different branches of sciences they worked. If by chance if you get time, you can just Google these names and you will find a lot of information. थोडा वेळ असेल तर हे करायला हरकत नाही आहे, पण प्रत्येक गोष्ट नीटसाठी मग नीटला त्यांचं लाईफ येईल का असले काही प्रश्न विचारू नका. मला तुम्हाला प्रयत्नपूर्वक या नीट आणि जेईच्या भ्रामक कल्पनेतून बाहेर काढायचं आहे. तुम्ही सायन्स आणि सायन्स शिका. एन्जॉय लर्निंग फिजिक्स, एन्जॉय लर्निंग केमिस्ट्री, एन्जॉय लर्निंग बायोलॉजी, and then enjoy exams. And if you are not enjoying physics and chemistry and biology, leave science. Leave science, don't take science. पण धीर सोडू नका. मी मगाशी माझ्याबद्दल सांगितलं, फर्स्ट इयरला मी जेव्हा होतो 1967, मला काहीही कळत नव्हतं, पण मला हळूहळू लक्षात यायला लागलं की खूप पॉवरफुल गोष्ट आहे सायन्स म्हणजे. And I got interested. I am with you today now. So please understand. So these are some of the basic discoveries done by these people. First cells were observed by Robert Hooke. Leeuwenhoek was also, but these cells observed by Robert Hooke were not true cells. They were all only the dead cells because they were only cell walls. Leeuwenhoek was the first to observe the living, moving cells, and we'll see in the PowerPoint presentation what kind of microscope he had. In fact, in the State Board book, that microscope is already shown. Leeuwenhoek's microscope has been shown in the State Board book. Robert Brown, he was the first to discover nucleus, because presence of nucleus, now we can usually observe the nucleus easily, but microscopy was not so well developed, and so Robert Brown clearly said that all the cells have nuclei. But then why special credit is given to Schleider and Schwann? Schleider and Schwann, they worked a lot and they tried to take different tissues, different animals and different plants and their tissues they observed. Schleider as a botanist and Schwann as a zoologist observed different materials, different plant materials, different animal materials. They must have observed sections of stem, leaf and root. Schleider and Schwann as zoologists, muscles and liver and kidney and whatnot. And every time they said that, yeah, every organ, every part of a plant and animal and fungi is made up of cells. So, because they worked a lot on this, special credit is given to Schleider and Schwann. That's what this has been kept in box. So, cell theory, actually credit goes to all, but usually cell theory is more credited to Schleider than Schwann. Ralph Virchow said, yeah, cells, pre-existing cells give rise to new cells, and Waldeer was the one to discover the chromosomes and so on. So I think these things we can once again look at. Looking at these people, and very interesting. Europeans, all of them being Europeans, Europe seems to have been the cradle of original scientific discoveries. Although now US, America, and Canada, and Australia, and to some extent China, they are all involved in research. India also to a very small extent. Okay. But basically, it was the Europe where all science started. So let's go for the PowerPoint presentation. Robert Hooke, a British person, but not at always exactly 1665. He was working in the field of science for several years, and this particular year which is recorded here is for that particular event when he published his research and showed that cells are there, and he actually observed the skin of a tree, dead cells of a cork of a tree, and that picture will be shown to you a little later on. This was a kind of microscope. He was a great person, very intelligent. Basically, he was a physics person. Physics people are always intelligent. Yeah, that's correct. That's very correct. So this is how this is the microscope which he used. But those days, he used an artificial light here, some candle or some kind of lamp, oil lamp to illuminate the object. So the object was illuminated by some kind of oil lamp or candle, because if you are working in the laboratory and if the light is not enough, then that was the solution. Okay, next one. This was the microscope I told you. This is Leeuwenhoek's microscope and this is Robert Hooke's microscope. 350 years back. So the microscope must, the kind of glass, the metal body, and all these things are wonderful. But this microscope is very simple. And in the state board book, this picture is given. Let me tell you, with this microscope, Leeuwenhoek was able to observe more better things than Robert Hooke. That's wonderful. So you don't need to have a complicated apparatus. You can have still a simple apparatus. Next one. These are the cells which Robert Hooke observed. These are the Koch cells. This is very famous. And these are the compartments. These are only cell walls because there is no cytoplasm. These were not living cells at all. He published many things, many books he wrote, and Micrographia is a very famous book. Minute bodies. This is all about his microscopic observations. Next. During those days and a little earlier to that, can you see this picture? This is from one of the European old type of towns, and this is just for fun, but this is an imaginary picture. You know, those days lenses were not necessarily available in the form they are available today, and all our specs, glasses are all due to the lenses. And you can imagine, this is an imaginary picture where there must have been some kind of a lens shop, and people, when they need lenses, they must have ventured into the roads to find out if there is a shop. And it must be a very difficult time. You know, we have specs and now everything becomes easy for us, but those days it was all beginning. So this is a kind of a shop, spectacle shop, you can say. Things are being tried here and there. Next one. Leeuwenhoek, a little later than Robert Hooke, but almost around the same time. He was a business person, and so he did not have any formal education of university type, but then he was always having a great interest in knowing things around, and he prepares tiny microscopes, just a copper plate and a pointer, and he manufactured 500 such simple pieces, and some of them are still there in some of the museum of the world. And it was a wonderful simple structure, a simple microscope with the help of which he could observe several things. Let's see which are those. Yeah, this was his microscope, and he prepared 500 such things to be distributed to people. Lens was fitted here. Can you see this one? This is the specimen for the pointer. Here he would take a drop of blood, or a drop of dirty water, or a drop of semen to see the sperms. And there was a kind of a screw, and then he could adjust the screw to focus the thing. So when this needle could be moved here and here to adjust it so that it could be focused. Wonderful. This was a Dutch person. Yeah. These are the drawings, original drawings which were made, and see the handwriting and see the beautiful drawings. They still exist in the books of those days. So next, I think we'll go back, please. Go back, please. Go back. I have a question to Kaustubh sir. It is Leeuwenhoek and also the Robert Hooke, and they have long hair. Does it mean that those days there were no hair cutting facilities?
No, those were wigs. They used to wear wigs those times.
Why Newton should wear a wig, and Robert Hooke and Leeuwenhoek should wear wigs? Today's scientists, they don't wear the wigs.
No, it was maybe it was some kind of style statement or status symbol.
Status symbol, right? Okay.
Judges.
Okay.
Judges used to wear.
Judges used to wear, and the scientists also used to wear. Because I always wonder why they have such kind of long hair. Oh my God, that's a good information. Let's go ahead. Okay, he is not wearing a wig. Robert Brown. So Brownian Movement. He was also interested in physics and lots of other branches of science. But then he was also, those days people did not unnecessarily differentiate between physics and chemistry and biology. They all considered all sciences to be one. And so a physics person interested in biology, a biology person interested in chemistry, that was very, very common. He was the one, and he was a little senior to Darwin, and he was a British person again, and he was the one to show that yes, cells have nucleus. So his contribution to the cell theory is that he was the one to point out that the nucleus is present. We'll go ahead then. Yeah, Schleiden and Schwann. Matthias Schleiden and Theodor Schwann, a botanist and zoologist, worked independently. They were all German. They worked independently, and they were trying to find out whether every organ of a plant is having cells. And this great physician, he was a physician, a physiologist, and he was at the same time researching those things. That's one good thing about the medical field of Europe and even in the US. Every doctor is a researcher. Every doctor in Europe and US, those days and now, are researchers. In India, that is happening now, but on a small scale. Some of our students who have become doctors, they have become researchers in that field. So if you want to go to medical, please make sure that you will enter into research. Research is not necessarily only for non-medical. In fact, there is so much of research in the medical, and therefore, to go to medical, you can although side by side do go into research. So this was a great person, physician, he was a physiologist, and then he was also a zoologist. He observed many tissues of the different animals and made slides and whatever microscopes were available during that time, and he continuously recorded those things, and he said yes, every organ has cells. Next one. Rudolf Virchow. Oh my god, this must have been imaginary picture. These people were great theorists, they were great academicians, and so he worked in different fields, and then he said that new cells come from pre-existing cells. Interesting. Great name, Louis Pasteur. He was the one. Microscopy was available in a better way. You can see the microscope here, and he was one of the most significant persons of that particular century, 19th century. He was a microbiologist, and it has been pointed out repeatedly that he was the one to find out many things. He worked in the field of enzymes, he worked in the field of vaccines, he made many vaccines. He was the first to show the cause-effect relationship between the germs and the disease. He was the first, along with Koch, Pasteur and Koch postulate, to say that diseases are caused due to germs. He was the one to work a lot in the field of fermentation. He can definitely be considered as the father of microbiology. Pasteur Institute is there at Paris even now in his name. Of course, he must have observed lots of microbes. There is no question about it. And when we are going to have a simple related practical for this lesson, you will be probably be able to see some of the microorganisms that we are going to grow in the laboratory. Oh yeah, Valdier, and he's famous for several things. Okay, so he was an anatomist, so fascia, glands, he was an anatomist basically, and he also discovered the chromosomes. Next. Huxley is comparatively more recent. He was in the 20th century, and he worked in comparison to the earlier pictures that you have seen. He worked in a better field. He probably had better facilities, and he was one of those people who tried to put together the old concepts and the new concepts. Tried to put together the cell theory and the genetics, the cell biology and the genetics, and other areas of synthesis. It is very necessary that somebody discovers certain things in genetics, certain things are discovered in cell biology, but all these things must be put together by someone, and he tried to put them together, and so credit goes to Julian Huxley for being able to synthesize science together. Whatever science is existing that time has to be put together to draw a meaningful conclusion. Well, we now go for cell, because ultimately out of 8, 5 lectures are going to be about the ultrastructure. So let's quickly understand what are the ultrastructure things that you have learned in the schools. Well, shapes of the cells are very simple. I think you know this: neuron and sperm cell and the plant cell and root cell, root hair, and pollen cell and red blood cells. Okay, that's, we have already considered this. We have already drawn those figures on the board earlier. Next one. This is the electron microscope. You know, when you want to learn the prokaryotic and eukaryotic cell in details, tomorrow we are going to learn the prokaryotic cell, and later on we are going to learn the details of the eukaryotic cells in 5 lessons, 5 lectures. Those are all going to be structures observed with the help of electron microscope. In India, easily there are about 100 electron microscopes now, but then earlier there were no electron microscopes in India. There have been great institutions in India also now. For example, Pune has a great place, NCCS, National Center for Cell Science. What is NCCS? National Center for Cell Science. It is located in the Pune University campus. We also had at Pune National Institute of Virology, NIV, and we have ICER, of course, and they share the facility of the electron microscope. Every institute cannot have electron microscope because it's a very costly thing, and to maintain and use it requires special abilities. Next. Here is a woman working on the electron microscope. Details of the microscope, electron microscope and other things, I think we are going to study in the 3rd part. Next one. Prokaryotic cell without nucleus and eukaryotic cell is with nucleus. We are going to deal with this in details. Next one. This is the typical animal cell. You can just read carefully the parts. Sometimes we ask questions to students. They say we are interested in biology. Then normally we ask a question. Yes, can you tell us parts of the cells? They will say, how can it be? If you are interested in biology and if your school books have already given to you some information about animal cell, plant cell and parts, and तुम्ही म्हणता आठवत नाही, मग कसं होईल? हे बघा, असलं काही चालणार नाही बरं का. नुसतं नीट करायचंय, डॉक्टर व्हायचंय म्हणून बायोलॉजी घ्यायचंय, हेच चूक आहे. तुम्हाला इंटरेस्ट आहे का? खरा इंटरेस्ट आहे का? खरा इंटरेस्ट फिजिक्स केमिस्ट्री मध्ये बायोलॉजी असेल तर अभ्यासाला लागा, आणि तुमचं सगळं शाळेची पुस्तकं कुठे टाकून देऊ नका, ते तुम्हाला उपयोगी आहेत. Each, all the boards, minimum level have considered the cell parts. Maybe in 8, maybe in 9, maybe in 10. You have to know this. Because when we learn cell biology now, we will have to assume that you at least know that there is something like cell membrane and smooth endoplasmic reticulum and rough endoplasmic reticulum and nucleus and the cytoplasm and Golgi apparatus, etc., etc. So you should know. But all these things cannot be seen with electron microscope at one time. असं नाही समजायचं की सगळं एकदा सेल्स खाल्लं की सगळं काही दिसायला लागेल. नो 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 नो. Special arrangements, special techniques are to be used to see the mitochondrion with electron microscope. Special techniques are required for endoplasmic reticulum. Very different technique will be required for nucleus. At a time, सगळं काही electron microscopy दिसत नाही. But electron microscopy, which started around 1950, made a lot of difference. Made a lot of difference in cell biology. Whatever cell biology we know today, probably it was not known to Robert Hooke. It was not known to Waldier, and it was not known to many of those great scientists like Leeuwenhoek and all. Because those days electron microscope was not there. Now we are able to see these things. Next. Plant cells, yes, similar things. Plant cells also have this schematic presentation of the electron microscopic structure of the plant cell. Most of the things are same, because there is a lot of similarity between plant cell and the animal cell. That was a little bit of the slideshow, and I'll just have some more comment to be passed, and then we will stop today's session. And just one small comment. Let's now make an end to today's session. Today's session is the first lecture of the first lesson of Standard 11 Biology. Feel excited about it. What is to be added today is something about dimensions. I think most of you are aware of these dimensions. 1 centimeter is equal to 10 millimeters. 1 millimeter is equal to 1000 micrometers. Please understand. Some of us know, some of us don't know. 1 millimeter can be divided, 1 millimeter, very small distance. It can be divided into 1000 units, 1000 small distances, and each small unit will be called as 1 micron. So 1000 small distances come together to make 1 millimeter. आपल्याला 1 millimeterच आपल्या स्केलवर दिसता मुश्किल असते. 1 centimeter तर ओळखू सगळ्यांना. 1 millimeter जरा काळजीपूर्वक बघावं लागतं की what is the gap between two lines of 1 millimeter, and that small gap between 1 millimeter divided into 1000 parts. How can it be possible with a microscope? So when you want to see anything which is at the micron level, micrometer level, you need to have a microscope. Please understand, for cells, this is the unit micrometer which is important. Many cells will be either small cells of the bacteria at 2 microns, or there may be liver cells which will be 10-15 microns, red blood cells suppose 7 microns, and plant cells are usually bigger, and parenchyma cells may be having a dimension of 50 to 100 microns and so on. So micron or micrometer is indicated like this. You can call this microns or micrometers. Please remember this. And it is very small part of a millimeter. Naturally, we cannot observe those small parts with naked eye. You need lenses, and when you are observing it with lenses, it is microscope. A single lens is a simple microscope. If you have 2 or more lenses, it becomes a compound microscope. And if you don't have lenses, and if you have electron beam instead of glass lenses, if you have electron beam and magnetic lenses, then it becomes an electron microscope. Please understand. So micrometers. 1 micrometer out of this 1000, 1 micrometer is 10,000 angstroms. I think many of you already have this idea of this very tiny measuring unit, angstrom. You know, many of the atomic dimensions are considered in angstroms. Many of the bond lengths between two atoms, bonds are there. Bonds are usually considered in the form of angstroms. Yes, you know, when you start thinking of the thickness of the cell membrane, cells may be suppose 10 micron, fine. But when you think of the cell membrane or the membrane of the nucleus, usually you will use the word angstrom because they are very tiny. So 1 micrometer can be 10,000 angstroms, and for convenience, it is possible to take 10 angstrom as a better unit, and that becomes 1 nanometer. Nanometer. So 1 nanometer is 10 angstroms. I'll use this. When you want to measure and talk about the diameter of the DNA, what is the diameter of DNA? Diameter of DNA is 2 nanometers. I'll just make a reference to this. This is the DNA molecule. And what is the diameter of the DNA molecule? That is 2 nanometers. That means 20 angstroms. So when you want to talk of these large molecules, or if you want to talk about the membrane of the cell, suppose here is a cell and you're talking of the membrane, what is the width of that membrane? Then it is in terms of angstrom. For example, the width of a membrane is 70 angstroms. So 7 nanometers you can say. 7 nanometers or 70 angstrom is the width of a membrane. Width of a diameter of a DNA molecule will be 2 nanometers, and so on and so forth. So I think for cell biology, you need to know the units micrometers and angstroms, and of course for convenience nanometers also. These two are the same. 10 angstroms taken together is 1 nanometer. Centimeter is not a useful thing except for very big eggs, ostrich egg and the bird's egg. Then they are one cell, but then they are in terms of centimeters. But otherwise, I think most of the measurement has to be at this level. So you should understand this. And most of the cells are between. Why 2? Because 2, usually bacteria will be of this size, 2 micrometers. Even the mitochondria will be of similar size. So a mitochondria in a cell is as good as the size of a bacterium. Bacteria are smaller, prokaryotic cells are smaller, and a eukaryotic cell is bigger, and the mitochondria and chloroplast are of the size of the bacteria. And some of the cells are bigger, and these dimensions can always be learned. Bacterial cell 3 to 5 micrometers, and a red blood cell 7 to 8 micrometers, liver cells 10 micrometers, human egg 100 micrometers. What is the size of human cell? 100 micrometers means 1/10 of a millimeter. 100 micrometers is 1/10 of a millimeter. Are we able to see the human egg? Because, by special procedure, human eggs can be taken out from the ovary. Are you able to observe it? Are you able to observe 1 millimeter distance? Yes. And 1/10 of a millimeter, that is 100 micrometer. Are you able to observe? You can observe. In fact, 100 micrometers is usually considered as the 100 microns or 100 micrometers is usually considered to be the limit of human eye. And when we will have a session on microscopy, that time we will talk about resolution, resolving power and magnifying power. We are able to see 1/10 of a millimeter. Not all of us, depending on our eyesight. 1 millimeter all of us are able to observe. But 1/10 of a millimeter, which is the size of the human egg, some of us can observe. But anything lesser than this, if something is 90 micrometers and 10 micrometers and most of the cells are very small, we are not able to observe. There is mention in both books, mycoplasma such as PPLO. You will read this carefully. Pleuropneumonia-like organisms. PPLO is pleuropneumonia-like organisms, mycoplasmas. They are the smallest cells and they are just 0.3 microns. 0.3 micron is lesser than, these are smaller than the regular bacteria, very small, 0.3 microns. So you should remember these things. So this particular sheet will be posted to you, and also side by side you have to go through the pages of both the books. Prepare well. फर्स्ट लेसन हॅज बिगन, त्याचा अर्थ अभ्यास केला पाहिजे. मग अशी काय मी सांगितलं तुम्हाला, रोज बायोलॉजीचा कमीत कमी 2 ते 3 तास अभ्यास केलाच पाहिजे तुम्ही, तर बायोलॉजी घेण्याचा फायदा आहे. आणि तसाच वेळ तुम्हाला फिजिक्स केमिस्ट्री द्यावा लागतोय, म्हणजे दिवसभर तुम्ही बिझी राहणार आहात. एक्सरसाइज करतात का? आम्ही सगळे करतो इथे. एक्सरसाइज केव्हा मी एकही दिवस वाया घालवत नाही. रोज एक्सरसाइज करा, अभ्यास करा. हे सगळ्यात चांगले दिवस आहेत एका अर्थाने, कारण आपल्याला शांतपणे अभ्यास करायला वेळ आहे. तुम्हाला दुसरं काही फार मोठी काही ड्युटी नाही आहे घरातली सुद्धा. थोडीफार घरातली तुम्ही कामं करा, पण अभ्यास करा. अभ्यास करायचा अर्थ वाचा, विचार करा, लिहून बघा. लिहिणं फार महत्वाचं आहे, हे फार मला सांगितलं पाहिजे. हे जे डायमेन्शन्स आहेत ह्याच्याविषयी खूप छान तुमच्या स्टेट बोर्ड बुकमध्ये हे बघा, खूप छान माहिती दिली बघा. सी या ठिकाणी काय काय मी तुम्हाला सांगतो. इथे ल्युएनहॉकचा मायक्रोस्कोप दाखवलाय. ओके, आय पॉइंट इट आउट दॅट देयर इज अ मायक्रोस्कोप बाय ल्युएनहॉक. हे बघा, सिंपल फॉर दाखवलाय त्यांनी, आणि हे खूप छान दाखवलंय. दिस इज हाऊ ॲटॉमिक साईज दाखवलाय. त्याच्यानंतर मॉलिक्यूलचे साईजेस दाखवले, प्रोटीन, कार्बोहायड्रेट, लिपिड्स, अँड देन व्हायरस, इन्फ्लुएन्झा व्हायरसचा साईज, मायटोकॉन्ड्रिया आणि बॅक्टेरिया. मी आत्ताच तुम्हाला सांगितलं ना, बॅक्टेरिया and mitochondria will be almost having the same size. Then the germinal cells of the animals and plants, and the frog egg, etc. etc. इथे दिले 0.1 नॅनोमीटर पासून 1 मिलीमीटर पर्यंत. So most of the cells are falling in this range, and these cells, eggs and all, are too large. निश्चित वाचा तुमच्या. So all the best for all that, and this session will be posted to you, and you have to start learning, and we will also be posting some questions, simple questions of about 10 marks. ते writing करायचं तुम्ही वहीमध्ये. Okay, that's it. All the best.